Welcome to Roadmap to Residency Evidence-Based Applications. Hello everyone, I am Abhiraz. Welcome to our Club 250 Matchmaker sessions. Uh, Here in this video, we will be basically talking about the reproductive system anatomy. We will not be discussing any questions. So whenever we talk about respiratory tract, we have to know that there is upper respiratory tract and then there is the lower respiratory tract. In the upper respiratory tract, it is due to the nasal cavity, pharynx, and larynx, and lower respiratory tract consists of the trachea, primary bronchi, and lungs. So we also need to know about the different zones of the respiratory tract. So there is the conducting zone and then there is the respiratory zone. So conducting zone is where there is no gas exchange and it consists of the large airways like nose, pharynx, trachea, and bronchi. Its function is to filter and warm and humidify the air. And it is also called as the anatomical dead space because there is no exchange of gas occurring in these areas. The respiratory zone is when there is actual gas exchange. So it consists of respiratory bronchioles, alveolar ducts, and alveoli. So beyond bronchi, it is called as a respiratory zone. And here, if in this within this respiratory zone, if there are some areas where there is no gas exchange, then that area is called as a physiological dead space, right? So now talking about the anatomy of the alveoli, which is typically important. So you can see the bronchioles give rise to the alveoli, and it consists of alveolar sac here. Okay, so these are the different alveolar sac, and these alveolar sac are connected to an alveolar duct, and this all collection is called as alveoli, and ultimately it all empties into bronchioles. So now talking about the lung anatomy as a whole. So here we have the larynx, the trachea, the superior or upper lobar bronchus, the right main bronchus, the middle lobar bronchus, and the inferior or lower lobe bronchi. And again, there we have the left main bronchus, and the bifurcation of right and left bronchus is called as a carina. And again, then we have the superior upper lobar bronchus, and then we have the inferior or lower lobar bronchi. We can notice that in the right side, there are three lobes, and in the left side of the lung, there are only two lobes. So that's why there is only superior and inferior bronchi, and in the right side, there is the superior, middle, and then there is the inferior bronchi. And again, the lung is then coated by the pleura, so the undersurface. So the immediate layer after the lung is the visceral pleura, and the layer after that is called as the parietal pleura. So visceral pleura is attached to the lung, and parietal pleura is not attached. So in between these two pleura, there is a space which is called as the pleural cavity, and which contains the pleural fluid. It's just like the anatomy of the heart and pericardial space. So another one thing that is very, very important are the lung segments. So the lung is functionally divided into different lung segments. So here we can see the right side, there is apical, posterior, anterior, and then again there is the medial, lateral, superior, and again there is the posterior basal, lateral basal, anterior basal. So now these all lung segments become important in different radiological investigations like the CT scan, the MRI. Then we can just label where the pathology is actually occurring. Instead of labeling a single lobe, we can label a specific part of that specific lobe. And this is called as a bronchopulmonary segment. And it indicates that each of these areas are supplied by each segmental bronchus. So we also need to know about the airway cells. So there are three main airway cells present. So they are the goblet cells, ciliated epithelial cells, and the club cells. The goblet cells secrete mucus, and they are more which is generally glycoprotein and water, and it protect against the particulates and infection. The ciliated epithelial cells are the beating cilia, okay, which move the mucus towards the epiglottis, and then it produces or the cough or that mucus is swallowed. The club cells which are present in the bronchioles are the non-ciliated epithelial cells, and they secrete the protective proteins, and they help in the detoxification from some smoke or something by P450 enzymes. Now, the histology of the respiratory tract is also very, very important and very commonly asked in the examination. The respiratory epithelium lining the upper cranial airways is classified as ciliated pseudostratified columnar epithelium. And this designation is due to the arrangement of the multiple cell types composing the respiratory epithelium. Now, while all cells make contact with the basement membrane and are therefore a single layer of cells, the nuclei are not aligned in the same plane, and hence it appears as the several layers of cells are present, and the epithelium is called as pseudo-stratified epithelium. Okay, now it is not like there are different layers of cells. This is the single layer, but this is one cell, and the another cell just extends up, and the nucleus is up. So that's why the nucleus is not lined in one layer, and it appears that it has multiple layers. That's why it is called a pseudo, okay, or false stratified epithelium. So this is a pseudo-stratified columnar epithelium with presence of goblet cells and cilia. Then you can understand that this may be a histology of the respiratory tract. Now, the majority of cells composing the ciliated pseudostratified columnar epithelium are of three types: ciliated cells, goblet cells, and basal cells. So the ciliated cells are columnar epithelial cells with a specialized ciliary modification. Goblet cells, so named because they are shaped like a wine goblet, are columnar epithelial cells that contain membrane-bound mucous granules and secrete the mucus, which helps maintain the epithelial moisture and traps the particulate material and pathogens moving through the airway. Here we can see the goblet cells which secrete the mucus, the basal cells here, the cilia. This is the basement membrane which upon the cell rests, and these are the capillary. So talking about the larynx, the histology of larynx. So larynx is an irregular tube connecting the pharynx and trachea. Now the skeleton of the organ is formed largely by a hyaline and small elastic cartilages that are bound together by ligaments and membranes. The striated skeletal muscles insert to the surface of the cartilages and provide to move against each other. The inner surface is covered by the mucosa, consisting of dense connective tissue with mucous gland and the eyelid of lymphatic tissue. Now, this is important. Now, the entire larynx is lined with pseudo-stratified ciliated columnar epithelium with goblet cells. Now, this is important information right there. The upper pair of the false vocal cords is covered by the typical pseudostratified epithelium, but the lower pair of the vocal folds consist of true vocal cords, which are covered by the stratified squamous epithelium. Now, this is an exception, so this is an important point to be remembered for the examination. The epiglottis closes the adytus into the larynx. Its basic tissue of the organ is the elastic cartilage. So the dense elastic cartilage is the epiglottis, and it is covered by stratified squamous epithelium on its lingual aspect, while pseudostratified columnar epithelium is on the laryngeal surface. The trachea is a tube about 10 to 12 centimeter long and 2 to 2.5 centimeter in diameter. The mucosa in the inner side is covered by the pseudostratified ciliated columnar epithelium with goblet cells, which secretes viscous mucus. Okay, and the submucosa is the layer of loose connective tissue with small mixed gland, and the fibrocartilaginous layer is a C-shaped hyaline cartilage, okay, and which posterior ends which is posteriorly free and ends are connected by ligaments. Muscularis trachealis is present in posterior part of the tracheal wall. Conducting zone. Okay, now here we can see the different zones, the conducting zone, the respiratory zone that we have talked in the beginning, and also the how the histology progresses. Here you can see in the trachea and bronchi, there is abundant amount of cartilage, right? And in the bronchioles, the cartilage is just disappears, and there is only involvement of the smooth muscle and simple ciliated columnar epithelium, not pseudostratified but simple ciliated columnar epithelium. Again, in the terminal bronchioles, there is the presence of cuboidal ciliated cells. So there is simple ciliated cuboidal epithelium. So pseudostratified ciliated columnar, simple ciliated cuboidal, and after that there is respiratory in the respiratory bronchioles, there is presence of the squamous cell, okay, and the smooth muscle is sparse, and there is simple cuboidal and squamous epithelium, but the cilia may also be present. Here you can see that the cilia is present all the way throughout up to the respiratory bronchioles, and in the alveolar sacs, there is present of type 1 and type 2 pneumocyte, the alveolar macrophages and capillary. Now, this picture to this slide is very, very important. Now, talking about alveolar cell types, there is type 1 pneumocytes and then there is the type 2 pneumocytes. The type 1 pneumocytes is of squamous in origin and it covers the 97% of alveolar surfaces. That it is a thin line of the thin line of the alveoli for optimal gas exchange. Type 2 pneumocytes is cuboidal and clustered, and it has two functions. It serves as stem cell precursors for two cell types, that is type 1 and type 2 pneumocytes, and it also separates surfactants from the laminar bodies. And the alveolar macrophages, they phagocytose the foreign materials and they release the cytokines and alveolar proteases. So that is the basic function of any macrophage. So the hemosiderin-laden macrophages, which may be found in case of pulmonary edema, in case of heart failure, okay. Now, these all are due to the alveolar macrophages. Now, the histological figure of type of the alveoli is very, very important, okay. And there is something called as the pores of Kohn. So the pores of Kohn are the anatomical communications between the alveoli that allow for the passage of air, fluid, phagocytes, and bacteria. The surfactant is very important structure and it decreases the alveolar surface tension, and it helps the alveoli preventing from collapsing. Okay, it maintains the integrity of that alveoli. Okay, and it also, since the alveoli is not collapsing, it also decreases the lung recoil and increases the lung compliance. It is composed of multiple lecithins, mainly dipalmitoyl phosphatidylcholine. Now, this term and this word is very, very important, DPPC. The synthesis begins nearly at 20 weeks of gestation and achieves maturity levels at 35 weeks of gestation. So, if there is a premature birth, for example, let's say at 32 weeks of gestation, then there is a high chance that the baby has not developed surfactant in an abrupt amount, and the baby can go into neonatal respiratory distress syndrome. The glucocorticoids is important for fetal surfactant synthesis and lung development. That's why in premature babies, if there is expected a premature birth, then glucocorticoids is administered prenatally or antenatally. Now, this is the basic histological figure of the alveoli. Now, let's talk about the lung development before we conclude this video. So, lung development occurs in 5 stages, which begins with the formation of a lung bud from a distal end of respiratory diverticulum during the week 4 of development, and it can be remembered as mnemonic. So, every pulmonologist can see alveoli. So for E, we have the embryonic stage, which is from the week 4 to week 7, where there is a development of lung bud, formation of trachea, bronchial buds, mainstream bronchi, secondary or lobar bronchi, and tertiary or segmental bronchi. Now, errors at this stage can lead to the development of tracheoesophageal fistula. Pseudoglandular phase, week 5 to 17, endodermal tubules, terminal bronchioles surrounded by the modest capillary network. Respiration is impossible and it is incompatible with life. In the canalicular stage, the terminal bronchioles develop into respiratory bronchioles, alveolar ducts, and it is surrounded by prominent capillary network. There was an increase in diameter. The pneumocytes develop starting at week 20, and respiration is capable at week 25. The saccular stage is when the alveolar ducts form the terminal sacs. The terminal sacs is separated by a primary septate. And alveolar is when there is week 36 to 8 years. So up to 8 years, there is development of alveoli. The terminal sacs from the adult alveoli due to the secondary septation. So in utero, breathing occurs via aspiration and expulsion of amniotic fluid, which increases the pulmonary vascular resistance through gestation. At birth, when we gasp for breath, the air replaces fluid and there is decreased pulmonary vascular resistance. From this table, they generally ask you from this notes part. Again, this is the same diagrammatic representation of the developmental of the lung. Thank you for watching. Keep studying hard.